Okay, sir.
Okay. So far we have discussed about your micro RNA databases, right? That is mir base. Well, now there is another class of RNA, an important physiologically important class of RNA called as LNC RNA. Okay. So, first we would discuss some biological aspect of this LNC RNA, and then we would check out what are the features that are present in this LNC RNA DB. Okay, so this LNC RNA stands for long non-coding RNAs. Okay, now this LNC RNA has a big size that ranges approximately from 100 to 100,000 bases. Okay, and apparently these long non-coding RNAs have no protein coding activity. Okay, so they do not code for any protein. Okay, now the interesting fact is that the number of these long non-coding RNAs are in similar number that are for mRNAs. I'm sorry. What I was discussing was key mRNA, the number of mRNA and the approximate number of LNC RNAs are of equal. We'll say, for example, there are approximately 20,000 mRNA as well as 20,000 long non-coding RNA. And based on this observed fact, then people started to make arguments that this LNC RNA might somehow be associated with expression of mRNA. Okay, and furthermore it was then later identified that these long non-coding RNAs are involved in normal development. They are also involved in regulating gene expression at various levels of development. Okay, and if these long non-coding RNAs are inappropriately expressed, that might lead to disease. तो ये well such type of publications and reports started to pop up in literature. Therefore, it was assumed that these long non-coding RNAs might be an important player in physiology of life forms. Okay, so now if you start comparing these long non-coding RNAs to mRNAs, you can end up comparing like these both are transcribed by RNA polymerase 2. Both of them, that is mRNA and LNC RNA, are spliced at certain locations. Both of these RNA types has got poly-A tail. Okay, and both of these RNA, that is mRNA and LNC RNA, has 7 methyl guanosine cap, but they only differ in the aspect that mRNA eventually codes for some protein, while LNC RNA do not code for protein. So some people also put forth the hypothesis that it might be possible that in due course of evolution this LNC RNA becomes pseudogene, and due to mutation at certain location, the ability of these genes or the ability of these RNAs to code for a gene was taken off, right? So and then different type of theories started to pop up in this area. Okay. Okay, so now as I have already mentioned that the number of LNC RNA matches to the number of mRNA. Okay, and that is where people started to speculate that these LNC RNA might be involved in, well, say for example, transcription and post-transcriptional gene expression. Okay, so well, say for example, as you can see over here in this figure, that you have got this mRNA that is shown in green are transcribed in the nucleus, and then these are transported outside where it is translated to certain protein. Okay. While on the other hand, LNC RNA, very few members are known to get outside the nucleus, right? So since they are confined to nucleus and the number of LNC RNA and mRNA, are of similar, have similar number, so it was then kind of estimated or postulated that they might be playing a role in transcriptional and post-transcriptional gene expression events. Okay. So, well, now what is that we know that this LNC RNA do in cell? So there are different groups that have demonstrated the functional significance of this LNC RNA, right? So I would be discussing some 8 experiments or 8 function that this LNC RNA are known to be doing in the cell. So the very first function that was proposed was that this LNC RNA, which might be present on say some upstream region, okay, upstream non-coding promoter, okay. So what these things can do is that they can negatively or positively affect expression of downstream gene that is shown over here in blue color. Okay, so it is entirely possible that such an event can happen by inhibiting the RNA polymerase 2 recruitment. Okay, moreover, since these LNC RNA are present in nucleus, and it is then it was postulated that this LNC RNA can induce heterochromatin remodeling. And due to the process of heterochromatin remodeling, well, it can somehow able to control the access to the genes of what genes can be expressed and what genes can be suppressed as such. Okay, so well, you can check out the details of all these events in this paper that I have cited at the corner over here. Okay, we lose it all in 2009. Okay, so another way this LNC RNA function in cells is through the antisense LNC RNA that is shown in purple color over here. Okay, so what happened is that this antisense LNC RNA is able to hybridize to the overlapping sense transcript that is shown over here in blue color for this gene which is present over here. Okay, so this gene when expressed, and it has got an antisense partner. So when this antisense LNC RNA goes and binds to some of the transcript of say certain gene, it might result in blocking the recognition of such splice sites, and therefore the spliceosome might miss out certain exons from that gene. Okay, and thus might result in some alternatively spliced transcript. So we'll say, for example, if everything went well, this particular gene that is shown in blue color, it should have created a transcript that has got 5 exon. But due to activity of this antisense LNC RNA, since some sites are blocked by recognizing by these spliceosome, the exon number 4 is missing in this alternatively spliced transcript now. Okay, so in this way, it can block the recognition of exon by the spliceosome, and thus it might result in alternatively spliced transcript. Okay, so in such a way, in such a state, if this antisense LNC RNA and the transcript that is present over here, if it is somehow recognized by the restriction endonucleases like dicer, it can generate the endogenous siRNAs. Okay, so this is also one of the possibility that siRNAs can also be generated in this way. Okay. So the next function that is known to be performed by this LNC RNA is by binding to a specific protein partners. Okay, they can also possess these RNA can also possess protein binding properties, and after binding to say certain protein, this non-coding transcript, that is we are talking about this LNC RNA. Okay, so it can somehow modulate the activity of protein by even by increasing its activity or decreasing its biochemical activity. Or if this LNC RNA goes and binds to certain protein, it might serve as a structural component that allows recruitment of other RNA protein complexes to form around itself. Okay, so in this way, it can play a structural or organizational role as well. That is, if it goes and binds to certain protein, it would send some signal and recruit some another protein to come and join the assembly. And in this way, it can perform these organizational role as well. The next thing that this LNC RNA can do is that by binding to certain protein, it can alter where that protein is supposed to localize. Okay, so it can alter the protein localization by binding RNA. The protein might end up at some different location than it should have been. Or these LNC RNA can be used to guide the protein as to where to go. Okay, so these are all the known function of LNC RNAs. Well, that either works at the transcript level or at the protein level. Okay. Now, last one is that this long non-coding RNAs, which are shown over here in pink, can be processed to yield small RNAs such as miRNAs, piRNAs. piRNAs stand for PV interacting RNA, and these PV interacting RNA are known to be involved in the epigenetic and post-transcriptional silencing of transposable elements. Right, this is also one of the interesting function that has been associated with this piRNAs and eventually LNC RNAs. Okay, and well, there are other less well characterized classes of small transcript that are still present, and it might play an important role in managing, maintaining, and regulating the pool of such small transcript that are not quite well characterized. So there is a lot of possibility that these LNC RNA can be involved in. Okay. So here is the list of some very important LNC RNA. So well, say for example, one of the most frequently discussed LNC RNA is hot air RNA. So this hot air RNA is one of the cancer-related long non-coding RNA. So this hot air RNA is known to form complex with polycomb repressive complex 2, what we call it as PRC2. Well, now this PRC2 is known to account for epigenetic silencing, and this epigenetic silencing is very frequent during different processes that include cancer development. So eventually this hot air is also one of the risk factor. Expression of hot air is one of the risk factor that can induce cancer in human being. Another important LNC RNA that is usually discussed is this exist and T6 pair. So this exist is a 19 kb LNC RNA, and T6 is its antisense partner. So this exist and T6 pair is known to modulate what you can say dosage compensation. Okay, what we call it as this pair is essential for X chromosome inactivation. Okay, that is dosage compensation. Okay. Now let us discuss some therapeutic aspect of this LNC RNA, since these are known to play an important role in disease. Okay, let's say for example in case of cancer, LNC RNA called as hot air, as we have already discussed it. It plays a role in initiation as well as progression of different type of cancer, which include cervical cancer and nasopharyngeal carcinoma. Okay, so since it is known that this LNC RNA can be a risk factor for cervical cancer and nasopharyngeal carcinoma, the levels of this RNA are somehow are somehow used as a potential biomarkers for various human cancers. So what are biomarkers? These are small biological molecules which can be considered as an onset or as a hint ki, okay, if this particular biomolecule is found in a cell, that means something has went wrong and it might indicate progression of say certain disease. Okay, so this level of LNC RNA is thus also applied as a potential biomarkers in detecting various human cancers. Okay, this LNC RNA is also associated with Alzheimer's. Okay, now we all know that this protein called as beta secretase 1 or base 1 is a very crucial factor or is a very crucial enzyme that is important in progression of Alzheimer's disease and its pathophysiology. Okay, so this base 1 has got an antisense partner called as base as I have shown it over in blue color. So what this LNC RNA do is that it kind of might get involved in regulating the base 1 mRNA and eventually the expression of base 1 derived protein in vitro as well as in vivo. So this antisense LNC partner of base 1 is one of the LNC RNA, and it might play an important role in progression of Alzheimer's disease, a neurodegenerative disorder. So, well, you can, read these papers. Well, say, for example, this paper published in Cancer Biology and Medicine. You can read it for the role of hot air in cancer, and the paper which is published in Nature Medicine back in 2008, you can consider this paper for reading in more details of what is the role of this LNC RNA in Alzheimer's disease. Well, that is where I have taken these conclusions from. Okay, so you are advised to go and read those things in more details. Okay, so now that was all about the biology of this LNC RNA. Well, now let us talk about the informatics aspect of this particular RNA molecule. So, well, let us talk about why it was found out necessary to be, create a separate database for this LNC RNA. So, the necessity begins with the initiation of the Phantom 6 project. So this Phantom 6 project, also called as functional annotation of the mammalian genome. So this Phantom 6 project was initiated to systematically elucidate the function of long non-coding RNAs in human genome. Okay, and by this effort of Phantom 6 project, people were able to catalog more than 30,000 putative long non-coding RNA transcript by year some 2011, that is some 10 years ago. Okay, so now in due course of time, the data started to accumulate at a very astounding rate. Okay, and these impose challenges in say molecular and functional characterization of long non-coding RNA, as well as it become increasingly difficult while maintaining the efficient catalog of how many LNC RNAs are being discovered and being published in the literature. So another important factor being that this LNC RNA data was not initially accounted in reference sequence database as well as Unigene and other related standard databases. Therefore, considering all these facts, it was it was figured out that there should be a separate database that would deal with only the information as well as sequences of this long non-coding RNA. And therefore, this database was established that include mammalian as well as eukaryotic species data, as well as detailed annotation of LNC RNAs, whatever it was available at that time. Okay, so the aims of LNC RNA Db was to facilitate or to act as a central repository of all known LNC RNAs in eukaryotic cell. The objective was to facilitate research of this poorly annotated gene, to decrease instances of replication of the same data, also to integrate with other resources, whatever LNC RNA data is available. The objective was to integrate with the external resources like UCSC genome browser and NRED, that is non-coding RNA expression database. Okay, so by integrating these LNC RNAs to external genome browser, like UCSC is one of the most popular genome browsing service, which people usually use very frequently. Dr. Abhijit Kulkarni will tell you, in more details of how do you use this UCSC genome browser. Okay, so hang on till your genomics lecture begins. Okay, so the idea was to integrate with other resources. Okay, so what we call it as proposing of this LNC RNA DB has enabled researchers to get interested in LNC RNA, and it become a convenient place to find wide range of information regarding genes of interest from a single location. So considering all these issues, LNC RNA DB was established. So now let us talk about what are the content of LNC RNA DB. Okay, so initially this LNC RNA DB only consisted of imprinted LNC RNA, LNC RNAs that are associated with certain disease, or these RNAs that were either pathogen induced or either pathogen derived, bifunctional RNAs, LNC RNAs, as well as the LNC RNAs which are yet to be assigned any function. तो ये सारा डेटा पहले उनके डेटाबेस में था. Okay, so let us talk about what are imprinted LNC RNA. So these LNC RNA function to control the expression of other genes. For example, air is known to air and KNCQ1010T1. Well, these are examples of these LNC RNAs that are known to control the expression of other genes. So there are disease-associated LNC RNA. So these are the LNC RNAs that has got tendency to express in disease condition. Well, we have already talked one, that is base AS, that is antisense partner of the gene base 1 that is involved in Alzheimer's disease. Another example that I would like to cite is in terms of HSR omega gene, which is involved in polyglutamine induced toxicity and neurodegeneration. Okay, while there are certain LNC RNA that can be pathogen induced or derived or pathogen derived LNC RNAs. We'll say, for example, some LNC RNAs like PINC-UP are produced and modulated by pathogen or host cells during the pathogen infection. Well, pathogens are usually known to hijack the cellular physiology and use the host cellular mechanism for their own progression and development of their life cycle. So it is entirely possible that some of the LNC RNAs, functioning of certain LNC RNAs can be hijacked by certain pathogen. Okay, so as I have already mentioned, this PINC1 is known to be upregulated during the plant infection. Okay, so not only humans but also in plants also it is involved in, say, certain disease conditions. Okay, these bifunctional RNAs have multiple independent roles. Okay, they can act as splicing isoforms that are known to encode protein, whereas if they are spliced in a specific way, they can encode for regulatory LNC RNA. For example, this LXRB codes for the protein, while if the gene is alternatively spliced, it can code for LNC RNA. Okay, so this is the best example. Then we'll say, for example, you have got LNC RNAs that do not have any function. Okay, like 4 antisense LNC RNA. Well, this RNA is not well described in the literature, but whose function, well, it is still yet to be known. So this is the wide range of type of LNC RNAs that you can expect to be present in your resource. Okay, so in 2015, a new version of this LNC RNA Db was released, and the new features of this second version was incorporating, of course, new entries. Okay, so some 283 entries from 17 different organisms were figured out, and their data associated with LNC RNA sequence was deposited over here. New interface was designed that was focused on tissue and disease association. Okay, then sequence search capabilities were also incorporated, like the blast service was set up in 2015 version, so that people can perform similarity search to detect if they have hits related to the given sequence or not. Okay, then this version also incorporated the gene expression data, that is expression of selected LNC RNA in 16 human tissues from human body atlas. Okay, this was one of the biggest advancement. And further data accessibility was improved by enabling the XML content of the entries. Okay, so if the data is stored in XML format, it becomes very easy and convenient for people. Sorry, it becomes easy and convenient for people to analyze using computer readability. Okay, and of course user submission capacity was also increased, which was not present in the previous version. So this is how the interface looks like if you visit the current version. Okay, so all you need to do is that you can put over here the gene ID or keyword, functions and species. Okay, you can also set a few filters, or if you have got a sequence, you can enter the sequence over here and just hit this blast button so that you can perform sequence similarity searches as well. Okay, so this is the resulting typical output of this LNC RNA DB. So what you can see over here is the main entry ID, accession number. You can download the XML page, and maybe you can perform some programming related duties or programming related stuff over here. Okay, so you have got this name and some synonyms or other aliases by which this particular LNC RNA is named. Then there is a section of characteristic that describe the LNC RNA. Then there is section of expression that keeps tab of known expression of LNC RNA where it is expressed. Like the function section describes the functions of this LNC RNA. This miscellaneous section has got miscellaneous information like anything that is related. Then as I have already mentioned you that it has been incorporated with human body at last, and due to this now it is possible to to determine at what different body tissues the expression of this LNC RNA is associated with. Okay, well, say for example, this particular LNC RNA is most expressed in WBC, while it is least expressed in adrenal gland. So you can have a idea of where exactly the expression profile might be working. Okay, then you have got section for species, okay, which deals with species and chromosome locations that are over here. These are links to the UCSC browser. Then there is a separate section for literature, wherein you have got the list of PubMed IDs that are also hyperlink. If you click over here, you would be taken to the PubMed, as well as a short description of this citation in terms of the name of author, what is the title, the year in which it was published. Okay, and moreover, you have got some associated components like molecules that are known to be interacting with LNC RNA DB. Okay, and you have of course got the sequence tab, which keep tabs of the sequence detail. You can select to show the sequence, to view that sequence in FASTA format if you wish, or download that sequence in FASTA format. Okay, so these are the typical features of this LNC RNA entry. Okay, so this is for your further reading. Well, I am citing here two papers. The very first version of this database that was released in 2011, and the second version that was released in 2014. Okay, so you can as both of these papers uh, are published in Nucleic Acid Research, they are freely available. You can download them, read about these LNC RNAs in more details and the resources, how it was developed, right? All those details can be found in these two papers. So I am also providing over here the link of this LNC RNA database. If you follow this URL, you can follow it to its website and start doing some demonstration. I'm also giving you the project of this URL for this Phantom project, which kind of initiated this LNC RNA DB. Okay, so this is it for time being. Okay, uh, now I think it's being a lot of things for you today. Okay, this is it. So I would stop the sharing now. Okay, there is one more announcement that Sangeeta madam would like to continue for the previous session. Okay, so don't she will engage you for the next hour or so. Okay, so so don't go anywhere. Okay, the class is not over yet, right?